in between each interval.
Oh, okay.
Does
Makes
that make
sense.
sense?
Yeah.
So for yeast, I multiplied them all by 60. I did them all in minutes. So mine might be slightly different, but I don't know.
Okay.
So are you guys still talking about 6, pre-lab for 6?
Yeah.
Okay.
Yeah. But we can move on. We'll just look
No,
at
that's
it.
okay. I mean, this is key, right? This is what it's about. This is what we're supposed to be doing. This is what the whole thing's about. So yeah, just so looking at the 6. So Addison, let me ask a question about this. Are you talking about the graph or are you talking about the top 3? Are you talking about the whole thing?
The top 3.
Okay, so listen, if you look back, I just want to make sure on the second page of the pre-lab reading, right? So on the first page, it shows the binary fission, cell division of that Staphylococcus or Bacillus, and then below it shows the generations, right? It shows I have 0 generations, but I have 1 cell, so I have 1 cell. And then in generation number 2, I have 1 generation and 2 cells. Generation 2, I take those 2 cells and they divide into 4, right? So it's that geometric progression. On the second page, it shows 2 different formulas. The first one is F equals S times 2 raised to the T divided by X, right? And so what this is really all about is just algebra. That's really what they're showing us. And they're saying, okay, in the case of E. coli, its generation time is 20 minutes. And then it goes through. So underneath F equals S times 2 raised to the 2x, Tx, it says where F is the final number of cells. So the letter F, no matter what we're looking at on any graph now or any formula, F will always be the total number of cells that we end up with. Okay, then S is the starting number of cells that we start with. So
yeah, I get this. I just didn't know which numbers to put in for which, like, is the final number of cells the 409,600?
For which, okay, listen. So that is actually a really good question. Don't look at the whole graph. Look at the first 2 sets of numbers. So for E. coli, yeah, the time in minutes equals 0 to 60. Okay, so the doubling time or the generation time for this one or the time that they're using for E. coli is how many minutes? Between each set of numbers, how many minutes are they using?
60.
60 minutes, right? So we know that 60, so it's going to be F equals, or it's going to be X equals, okay, and the above number is going to be 60 minutes for E. coli, okay? So for T, so X equals 60 minutes over 3.3 log. And now we got to figure out what F and S is. So F is the final number of cells that we end up with. So looking at that first 2 sets of numbers, 0 is the starting time, 60 is the ending time. So what is the total number of cells in that generation that we end with? That's the final number of cells we get.
800.
There you go, 800. And so it's going to be 3.3 log 800 over something. Well, what's the S time or T?
100.
Okay, yeah. So what? Yeah. So exactly. So it's 800 over 100. So the formula is X equals 60 minutes over 3.3 log 800 over 100. Okay. So before we move on, now it's the algebra part. So if I'm doing this by hand, which part of this do I want to do first? The math part. If I'm doing each step by hand, what would I do first? Would I go 60 minutes divided by 3.3 log? Would I go 800 divided by 100? Which part of that would I do? What was that?
That's what I would do. 800 divided by 100.
Okay, so 800 divided by 100 is what?
Isn't it 8?
Yeah, it's 8, right? So now I've got 3.3 log raised to the 8th power, and then I'm going to take whatever that number is and divide it by 60. So now what's your next order of operations? What am I going to do next?
So can you not just plug this all into your calculator?
There is a way to plug it all into your calculator in one shot. And all you have to do is you got to go, okay, so when I set this up into my calculator, I go 60 divided by, and then I put a bracket and I say 3.3 log. I put another bracket and I go 800 over 100 and then 2 brackets to end it. I'm telling my calculator, I want you to do 800 divided by 100. I want you to do 3.3 log raised to the 8th, because it's going to do that, and then it's going to do 3.3 log next. And then last, I want you to take that number, and I want you to divide 60 by it. And that's the number of minutes that you'll get. In the case of E. coli, it's going to be 20.13 minutes when you do the math. Now, let me ask you something. In the notes and the number of generation time, what is the generation time for E. coli without doing any of the math? What is the generation time for that? Well, if you look at the notes, it tells us in the notes right here at the very beginning of this lab. Okay, on page number 2 of the pre-lab, it goes down and it says, whereas E. coli can double in as little as 20 minutes. So E. coli's doubling time is 20 minutes. Well, when we did our math, what did our math say it was? Our generation time was 20.13 minutes. So we know for that one, we know we're right, because our generation time that we calculated and the generation time that all of our books and everything tells us is dang near 20 minutes. Okay.
Mm-hmm.
So let's look at the next one really quick. So looking at yeast, forget the units right now. So what is the generation time for yeast? So take the top line, and what's the time below 0? The first line. So for E. coli, what was it? It was 0 and
60.
60. So for yeast, it's going to be 0 and
120, or is it 2?
It's 2, because the units is what are the units for yeast that we're going to use?
Hours.
So for whatever reason, the scientist doing this study is using hours, okay? And we don't know why, but what I'm saying is, because the data table gives us hours, we're going to look at an hour. And what Laura was trying to say to you is, Laura did what? She converted these times to all minutes. I think that's what the discussion you were having, right, Laura?
Yeah, that's
Hello?
right. Yeah.
Okay, that's all she's saying. So what I'm telling you, Addison, don't do that, because this data set and the instructions do not tell you to convert it. Just take x equals 2 hours over 3.3 log. Now tell me what your generation cells is. How many cells for yeast do you start with, and how many do you end with?
So you 100 and you end with 200.
You ended with 200, and what did you start with?
100.
Okay, so you tell me what's F and what's F and S. What am I gonna, what over on the raised? What am I gonna raise to what am I gonna raise?
2.
So I'm gonna, how do you get 2? You tell me how you got 2.
200 divided
200 divided by
100.
It cut out on me. Okay, and then when you divide that, what did it give you? It gave you 2. Awesome. Okay, and then from there you just finish the math. Okay, so then you'd have, and then your unit time. So now the reason this is important is so on this one here, your math says that you got 2.013. What's your unit time for yeast? What unit did the book give us? Did the instructions give us for yeast?
I don't know.
Okay, so the time that you started with for yeast cells was hours. So for yeast, because we calculated it, it's 2.013 hours. Okay, and that's, you got to, again, you always have to maintain your units unless it specifically tells you to convert it to something else. That makes sense? Okay, looking at the last one, without any help now, you tell me how you're going to set up the mouse fibroblast. You tell me how you're going to set that one up.
So 2.3 400 divided by 1.
Okay, and what are your units? On the top line, x equals 2 what? What's the units going to be?
To the 4th.
No. X equals 2, and is it hours? Is it days? Is it minutes? So write days next to 2, so you know that when you're done with your math, it's going to be a number to days. The scientist in this wants to know the fibroblast doubling time, and the amount of time that they're using for their units is days. So it's 2 days over. Finish the end. What's the bottom line say?
Over. I don't know what you're saying.
So 2 days is over what? What was that?
Okay, just a second.
Yeah, no, I'll wait. Yeah, no, I guess I just want to make sure that you can look at these data tables and you can take that formula and find the numbers because, like, again, on the exam, that makes sense. You can be able to pull these data sets apart. But no, you're getting it. Is that okay? The only reason I spent some time talking about it is because this data table sucks because they change the units on you each time. E. coli, they're asking for it.
Decimals places did you round?
Oh, I just did what it didn't say. Again, if it specifically said it wanted significant figures, my numbers would be a little bit different. But so like in E. coli, I did 20.13 minutes. And that's just what but my decimal was longer than that. I just cut it at 3. I said screw it. And then I did 2.013 for yeast because again I just got lazy and didn't want to do any more. The numbers were longer than that. And then the mouse fibroblast, it's 1.006. And I cut it there because it was like 006 is rounded down, it's gonna be 1, it's gonna be 0. And then in units is days, so it's 1.006 days.
Okay, thank you.
Does that make sense? Okay, and here's the thing. Yeah, just if you chose, like, for time of yeast, if you chose 60 and 120 as the time that you were doing your data set, like that you were using, then you'd use 800 and 6400. Guess what? You'd still get 20.013, no matter how. Or if you went 0 to 120, then you'd go 0 to 120. You'd go 100 to 6400, and your unit time for your doubling time for that would just be 120. Your top number on the top would be 120. And if you do that, you're still going to end up with 20.0113. No matter how you set those up, you're going to always end up with the same ending number. At least you should.
Okay.
Okay, I'm going to shut up. I'm sorry. I beat that. Okay. Ready to move on? Ladies, everyone, Laura, everyone?
Yeah.
Yeah, I'm ready.
Okay. So we're on chapter 13.
Sorry, really quick. What was the generation time for the last one?
For the mouse stuff?
No, for the one with the table.
Okay, so the generation time I got was 23.188 minutes.
23.188?
Yes, minutes.
And is that, what did you do for that? Did you do 30 over 3.3 log? And then
so that one there.
60?
So the generation time. So on that one there, generation time is where the actual cells are doubling. So the cells don't start doubling until 90 minutes. So at 90 minutes, that's when we see our log time start. So I went X equals 120 minutes, and 120 minutes was 210 minutes. So at the 210 minute mark to the 90 minute mark, that's when our doubling time is. So if you look at it, you've got 0.02 to 0.05. That's doubled. 0.05 at 120 to 50. So if you doubled 0.05, you'd get 0.10. Like if you timed it by, times it by itself, it'd be 0.10, but we got 0.12, so it's clearly doubling. Okay. 0.12 at 180 minutes becomes 0.30. That's doubling. So basically what you're looking for is where it's doubling. So it's 90 minutes to 210 minutes. And then it levels off at 0.74. It goes to 1 at 240, and then it's 1 again at 270. So then we're at our stationary time at that point. Okay, so our graph goes up. So it's X equals 120 minutes over 3.3 log. And then our ending cells is 0.74, and our starting number of cells is 0.02. So you go 0.70 divided by 0.02. That's going to give you the number. 3.3 log of that number raised to that number, and then it's 120 minutes divided by that, and I got 23.188 minutes.
Wait, so what's your F and S?
My F and S. So
yeah.
So again,
I was following you with the where it doubles.
So look at on the table above. It says percent of absorbance A to A 600, and it's in that quotes. That's the number of cells that are being created in each generation. So look at at 30 minutes, how many cells did we create? How many?
0. So you started at 90 minutes with the 0.02.
Yep. And then I went to now. Where does the doubling end? So 0.030 to 0.74 doubles again, right? But
Yeah.
from
So are you saying it ends at
0.74
240 or 210?
210 because it starts
Because
to
it's
drop
not
off.
quite doubled.
It doesn't double at 240. So at 210 is the last data set that it doubled, and then at 240 it didn't double. It was only a quarter of the growth, and then again at 270 it didn't grow at all. At that point, we're clearly in our stationary phase.
So your F was 0.74 and your S was 0.02?
Yes.
Okay. All right. Now I get it.
Okay.
Thanks.
Good.
Yeah.
Okay. Are we all stable and everybody kind of gets a feel good about that?
Mm-hmm.
Okay, and then Addison, when I see you on Wednesday, we'll go over it. I'll make sure you understand it again.
No, I got it.
You got it?
Yep.
Okay. And then just really quick, you guys, did you look at the practice sets? So in chapter 9, she sent out some practice numbers. She sent some stuff out. So like, for example, she said, yeah, she said, so she talked about today, if you start with 6,000 bacteria cells and the generation time or doubling time is 30 minutes, what is the number of bacteria that you'll get in 90 minutes? And the formula for that is NT equals N0 times 2 raised to the T over D. Okay, so she uses a little bit different formula, but it's the exact same thing that we're getting. All she's asking for is this one. If she's flipped it, she's not actually for the generation time. We know that. She's saying, hey, how many cells do I get? And so she's saying, hey, given this many cells, here's your generation time. And then she gives you the total number of generations in 90 minutes. Well, what's 90 divided by 30? That's 3. So it's 2 raised to the third times 6,000 bacterial cells. That's going to be, and then you times that. That's going to be your total number of cells at the end on this one, that first example. So I'll go over it with you guys on Wednesday if you want the actual examples from chapter 9 that she gave us. Mm-hmm. But it's pretty, I mean, you guys, I know you guys can do it. It's just knowing what the heck you're looking at, and it's pretty straightforward.
Okay.
Okay, we cool? Laura, you good?
Yeah, I'm good.
Okay, so moving right along. Oh, ladies, just so that you know, Addison knows this already. I can actually record these and put these on YouTube, on my YouTube channel. And so today I'm actually recording this and it's going to go on my YouTube channel. So if you wanted to review what we're going through, you can go back and find this on my YouTube channel and you could go through everything. You hear everything we're talking about. And so if that bothers you, just let me know.
Oh, that's good? Awesome. Thanks.
Yeah, so you can go back and see what we've done with our voices, with the slides that we looked at, and you can beat yourself to death with everything we were doing. Okay, so we're on chapter 13. So again, without looking at the slides really quick, what do we remember from chapter 13 today? Just looking at the title of this particular chapter.
Like disinfectant stuff.
What was that?
Like sterilization.
So yeah, so what was sterilization? What was that? What is sterilization?
Like clean all the basically.
Yeah, so it basically you're killing everything, right? Mm-hmm. And so in unit 9, we were talking about doubling times and how fast stuff grows and everything. In this chapter, we're talking a little bit more about actually killing this stuff or slowing it down. And why is that important for us? For those of us going into healthcare, why is that important?
Clean.
Well, okay, so I'm dealing with two things. I'm dealing with people and I'm dealing with stuff, right? Okay, so let me ask Laura, since Laura's on mute, but Laura, listen, and then you can answer it really quick.
I'm listening.
So Laura, why exactly do you wash your dishes at home? Why don't you just eat off dirty dishes every time you eat?
Because you don't want to get sick, so you want to kill the harmful bacteria.
Yeah, exactly. We clean stuff to keep us from getting them, right? Okay. Now, the other side of this is, now, Addison, you and I did pathophysiology. We didn't talk so much about us getting sick. We talked about already sick people. So what were some things that we can do for people who get bacterial and viral infections? They already have them. What do we do for those people?
Use precautions.
Yeah, like, so we take precautions so we don't get it, right? And then we also get treatments going. Like we start to do treatments for people. So let me ask you guys a question. We talked last time, Laura, you and I think I spent a little bit of time talking about this. What if we have a bacteria in somebody that really doesn't like oxygen, that oxygen kills it, would that be a wound? If say they got a cut on their leg and we did a culture and we found the bacteria causing the infection really doesn't like oxygen. Well, what is one of the treatments that we can give to somebody with a bacteria that doesn't like oxygen?
Did we talk about that? I just remember if it doesn't like oxygen, it's going to stay away from the outside of
Yeah,
the
so let me use a good
ring.
example. I got somebody with some raging gangrene, and that gangrene, it's this nasty wet gangrene, and it's starting to die. And I find that the cause of the gangrene is a bacteria that doesn't like oxygen. Is that a wound that I'm going to bandage up and put a lot of Vaseline on?
Oh
And
no, you're going to keep it open.
yeah, you're going to want to irrigate that, clean it, and let that be open to the air. You may even actually put like an oxygen blow, like you may actually have a tube with 100% oxygen blowing onto that wound. Why would I do that?
So that you kill it because it doesn't like oxygen.
Yeah, you're creating an environment that thing does not like, and that the environment's going to kill it. And then, oh, by the way, now we add an antibacterial medication that we're going to use, and now we've hit this thing double whammy, right? If it tries to go further down into our tissues, the antibiotics are going to get to it and kill it. And then, oh, by the way, so it's going, well, I can't go deeper in the tissues because there's antibiotics killing me. And so what does it try to do? It tries to move away from that. Well, now you're blowing oxygen to the other side of its environment. It's like, so as it tries to surface to get away from the antibiotics, the oxygen on the other side's killing it, and you give it a double whammy. Does that make sense? That's why what we're learning in chapter 13 is so important. It lets us know, hey, man, I got this thing that likes carbon dioxide. You know what I mean? I got this thing that doesn't like carbon dioxide. I got this thing that loves carbon dioxide, right? Okay, we cool? Okay, I'm done. That was just, again, so if you look at everything she talked about in today's lecture, pretty much, it was, I have stuff, inanimate objects, and I have live tissue. And then how they, and the bacteria that grow and have an affinity for those areas, and then the names that she used for those things, right? And that's it. So again, as you're kind of breaking this, okay, I want to control microbacterial growth. Well, am I trying to grow it in? Am I trying to control it inside of live tissue, or am I trying to control it on the silverware at my house? Well, the silverware at my house, I can do things to silverware that I can't do to the human body because I can't kill silverware, but I can kill a person trying to kill the bacterial growth. Okay, ready, go. Okay, chapter summary. We kind of just did the chapter summary, so I'm going to shut up again. Okay, here it is. Okay, here we go. Here's the keywords. Addison, you hit the first one.
Okay.
So you want to give us the download on the first one? What are the key things for sterilization?
Removal of all viable organisms, inanimate objects.
Okay, so is this for people or is this for silverware at home? I didn't hear you, Killer. Yeah, every other word. For some reason, sometimes you cut out. I'm not sure why. We hear every other word you're saying, and then you finish talking. So sterilization, disinfection, and sanitation. These three terms deal with what thing?
Inanimate object.
Yeah, this is for so Laura's silverware, if she doesn't want to kill her husband, she wants to sterilize, disinfect, and sanitize her silverware at home because she would like to not hurt her husband and herself. And so so killing everything is sterilization. What's disinfecting? We're only killing what and disinfecting?
Pathogen.
Yeah, we're just killing the stuff that's harmful to us, right? We're only interested in that. Okay, and then sanitation. What's sanitation? What's going on with sanitation? Are we killing anything in sanitation?
No, you're not.
Yeah, we're just decreasing the total microbial population in sanitation. We haven't really killed everything. We just we're going to sanitize it and get it relatively clean. And the reason that we do that is because we found that at certain levels, some bacteria at certain levels they're safe around humans. They don't cause us to get sick, so we don't have to kill it. Where sterilization, we're killing everything. Nothing's surviving that. And then disinfecting, we actually target pathological organisms. We're trying to kill all those. We don't kill if we kill anything else. And then sanitation just says, good is good enough. Okay. Now, these three words are dealing with what?
Living tissue.
Humans.
Yeah, these are all
Yeah.
living tissue. So so let's look at the first term. Hey, Addison, if I cut the anti off, what's this word right here? SEPSIS.
Sepsis.
What is okay, so what is sepsis?
An infection.
From pathophysiology, there is a kind of shock called septic shock.
Yep.
So if this is antiseptic, we're trying to keep this person from having out of control microbial growth, because if that microbial growth gets out of control, what happens to the living tissue?
They'll die.
Yeah, they'll die because they'll go into septic shock.
Sepsis.
Yeah. Okay, so antisepsis. Okay, so we kill or inhibit pathogens on living tissue. Yay. And then chemical agents that kill or inhibit growth of pathogens when applied to tissue. So can someone explain to me de-germing? That just sounds funny to me.
You're not really harming it. You're just making it leave, right?
Okay, so
You're just removing it,
what would be a good example then of de-germing?
like washing your hands. You're
Yeah,
not killing anything. You're stopping its growth. You're just getting it off of you.
So you're just removing it from one thing to the water and flushing it down the drain. Okay, so yeah, just de-germing. So from now on, Laura, guess what you can say to your husband? I want you to go de-germ the dishes. You can go de-germ sweetheart, go de-germ the dishes because I want to have antiseptic dishes. I want you to get rid of kill all those pathogens by de-germing them.
That's great.
So now that's a way to remember this though, right? If you said that, so Addison, next time your mom asks, so when I go shovel my parents' sidewalks, I'm de-germing them of snow. I'm mechanically removing the snow from there. I didn't kill the snow. I didn't get rid of the snow. I just moved it to the grass, not the sidewalk. Okay, then what's chemotherapy?
Kills basically all of them.
Well, and what's the key word in chemotherapy? So if I cut this apart into its two root words, chemo is what and therapy is what? The word chemo is chemical. So this is chemical treatment of something. So cancer, when we do chemotherapy to cancer, we're using chemicals. What's the difference between chemo and radiation for cancer treatment?
Radiation uses like, or I don't know what they're.
It uses gamma radiation and alpha particles. Yeah. They subject the tissue to high energy, to those higher levels of electromagnetic energy we call radiation. We're not introducing chemicals to their body. We're bombarding the tumor and the tissues with radiation. In chemotherapy, we're actually introducing a chemical that physically goes in there and kills the stuff. So that's what it intentionally kills or inhibits. So let me ask you guys a question about chemotherapy, whether it's cancer or bacterial or viral. What's the problem with chemotherapy in humans? What's the problem with this?
It kills all cells.
Cells too.
Yeah, it's really not selective, is it? Like if I introduce ammonia to the human body to kill a bacteria, well, is ammonia good for us? Do we like it? Do our bodies like ammonia? No, ammonia is basic and it's poisonous, and our body turns it into urea and gets rid of it. So if I start introducing excess ammonia to kill bacteria or viruses or cancer or something, then I'm also killing living tissue because our body has to now go through and clean up that ammonia, and that ammonia doesn't care, and our bodies just want to get rid of it. And so it's this kind of fight. And that's why people get really, really sick from chemotherapies. You're introducing poison that kills anything in its path. And then, oh, by the way, now our body has to work extra hard to try to get that excess poison out of it. Okay, so let me ask you another question. So some people choose to drink ethyl alcohol in the form of vodka mixed drinks on Saturday night. What they've really done is they've introduced a chemical into their body that is killing certain cells. They're doing chemotherapy. And what's the body's response to excess ethyl alcohol? What do people feel like the next day when they've introduced excess ethyl alcohol into their bodies?
I want to throw it up.
Yeah, they throw up, they feel sick, they have a headache. Like there's all kinds of, so that's chemotherapy that people are doing to themselves. They're intentionally introducing something that's poisonous, and that stuff is going through and killing or inhibiting the growth of other cells in the body. In that case, the therapeutic response is the inhibition release. It's more psychological and social. We're not doing it for an actual medical reason. We're doing it for a recreation reason. And then the payoff on the back end, the product is the hangover. Okay. We cool? Okay. Here's the common protocol. We'll just pass this. We can all look at this on our own. Okay. Who can explain what's going on here on this slide? Slide number 6.
So the sterilization kills the most, and the antisepsis kills the least.
Well, yeah, at the end of the day, they're all going to end up killing. They're all going to end up down here at the bottom, right? But which one's the most aggressive?
The sterilization.
Yeah, because look at what happens in the first, like the shortest amount of time, you have this huge drop in the number of bacteria that are there, or the number of microbes that are there, where disinfection, I'm still going to get a lot of death, right? We're still going to kill a lot of this stuff, but it's a little bit slower, and then it takes off, right? And then sanitation and antisepsis, I got a really, 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 really slow, and then bam, they get killed. It starts killing stuff. So remember how she talked about when we look at the instructions on, so like say you're using Windex to clean your windows and to clean surfaces in your house. And she talked about there's a D time, right? So if we're using sterilization techniques, the D time is quite a bit slower on the front end, but on the back end, our D time for whatever chemical we're using is going to be slower, right? So this one here maybe this is in minutes. So I got 1, 2, 3, 4, 5, 6 minutes, 7, 8, 9, 10, 11, 12 minutes. This whole time is 12 minutes. So a sterilization chemical that I introduce, I have to wait 6 minutes to get it to really kill a lot of this stuff. Antiseptic techniques, I got to wait till about 11 minutes to get a really nice destruction of the stuff. And then disinfecting, I got to wait about 8, 9 minutes before I can wipe it off to get the good death that I'm looking for to get down to this level. They're all a little bit different. Okay, we cool? Okay, here we go. This is where we get cidal and static. Okay, again, this is pretty straight up. So bacteriostatic and bacterial cidal. Well, that's bactericidal. So static, right? Like in the military, we say, hey, I'm going to do a static defense. Static means all I have to do is hold this ground. I don't have to kill the enemy to do this. I just have to stand on this line, and the enemy can't get behind my line. And then cidal, right? Cidal means I'm going to go out there and find the enemy, and I'm going to kill them. Okay? I'm going to actually go out and look for them and kill them. So bactericide, fungicide, viricide, algicide, those are chemicals or things that we're doing that actively go out and look and kill this stuff. Where bacteriostatic, fungistatic, it's like, listen, as long as this stuff doesn't grow anymore, we're okay. It's going to be fine. So what would be a really easy way to remember these? What's a mnemonic that we could think of that could help us remember these?
Well, she said suicidal.
Yeah, suicidal means
static. Just means like flat line.
Yeah, that's static. You're not going anywhere. Okay, or you can remember
the radio inhibits your ability to hear the music.
Yeah. Okay, that's a good way to remember it too. Or like I like these little bottles, these little like medicine bottles. Bactericidal standing on a bunch of dead stuff. Bacteriostatic is just standing on a bunch of little guys that are been arrested. They got the handcuffs on them. So, okay, this is the fun part: rate of microbacterial growth. So everyone, if you haven't remembered this, remember this is where there's the math. There's some more math now. In chapter 9. Generation time. What was generation time? Was generation time more cells or less cells? In chapter 9, we're talking about
more cells.
Yeah, we're talking about getting more and more and more bacterial cells. In chapter 13, are we talking about more cells or less cells?
Less.
Now, if you guys were watching today, all we're doing is working in reverse of what we've already learned. So don't freak out. We're just talking about the death or the limiting of growth of already established bacterias. And so really all we have to really do is reverse some numbers on the the formulas that we already know, right? So like, hey, for example, we're going to flip the total number of cells with the total number of cells when we end, and we flip those, and then we get a negative exponent in that spot. If we get a negative exponent, it's going to be a negative rate of growth, right? Does that make sense? And so just remember that algebraically, we're doing the math on these. That's really all she was talking about today, is just take the formulas you already know and flip them, and instead of getting a positive growth, you'll start to get a negative growth. And that's what she was talking about, okay? So microorganisms are not killed instantly. It takes time to build up this death rate, just like it takes time. You know, we have the lag phase, the log phase, the statutory phase, and the death phase. On the growth rate, we also have the same thing happening on the death rate, is that you're going to have a slow start to the death, and then it's going to just take off, and they're going to die really fast. Okay, population death usually occurs exponentially. So again, it's going to be that same slow and then rapid destruction, doubling each time. They're going to keep doubling down until they're all dead. Okay, and then D value was the decimal reduction time. So the time to kill 90% of the population. So 0.90 is going to be, because when you convert 90% to an actual number, it's just 0.90. And then here's the graph that she showed, which again, do you guys have any questions about this?
No.
Okay, there's one thing I want you to see. So there's the logarithmic scale and there's the arithmetic scale. So the red line is the arithmetic line, and the logarithmic scale is the logarithmic destruction of the cells. Can you tell me, why is the red line different than the blue line? Why is it different?
I don't remember, actually.
Let me ask this question. So how many cells did we start here at the very top? Number of cells starts at 10 to the 10, right? What do we end at for both lines? Both the red line and the blue line end where?
0.
At 10 to the 0, right? Which would be 0, correct? It ends at 10 to the 0, which is 0. So let me ask you this question. So I have the D value. The D value is an average cell destruction from time 0 to time 50, where the logarithmic scale, it doesn't look at it that way. It's looking at the cell destruction in 5-minute increments. So from 0 to 5, I got 10 to the 10 to 10 to the 9th on my D value scale. But on my logarithmic scale, how much cell destruction did I actually have from 0 to 5 on this graph?
Like 90%.
90%, right? So again, we have to be really careful when we're looking at these, and be really careful in the instructions, because the instructions might show you a big graph like this and say, yeah, okay, tell me your D value for this graph, and you calculate your average. And then it might say, hey, well, what's your real cell death from 5 to 10 minutes? Well, from 5 to 10 minutes, it's actually the D value at that point, because see that those actually make sense. They're like close. See, the D value is 10 to the 6th to 10 to the 5th. We're down here from 5 minutes to 10 minutes. It's 10 to the 1st to 10 to the 0. So the D value and the logarithmic value actually match at 5 to 10 minutes. That's the only place that they're the same, right? The level of cell destruction at that point is exactly the same at the D value, because they match. But everywhere else, they're not the same. And D value is over the whole time frame of the graph, where logarithmic, it cuts it up into these 5-minute blocks, and it's 90% each time. Okay, did I beat the math up again for you guys?
No, that was fine. That was like her thing that she had up on the board with the 90.
Yeah, all she did is she showed these numbers on the side here. She showed these numbers. She wrote this part of this graph out. She kept the did she have the graph up, or did she show this? I can't remember.
I think she did both.
Okay, here's the nasty surgical instruments. Okay. So let me use a good example of this. So it says toxic conditions influencing the effectiveness of the antimicrobial agent activity. So she talked about population size, okay. Let me ask you this question, ladies. Okay. If I have one pair of dirty socks sitting in the middle of a room, does that make the whole room smell bad?
Does that make
the whole room? Like if you have dirty clothes, like one pair of dirty pants sitting in the middle of a large room, sitting on the floor in the middle of a large room, can that one pair of dirty pants make the whole room smell bad?
Yeah.
It can, right? But does it get like progressively stronger as you get closer to the pants?
Yes.
Yeah.
Okay. What happens if I now add 10 more pairs of dirty pants to the same size room? What's going to happen
in your room?
Yeah, it's gonna be stronger smell. It's gonna happen faster, right? Okay. So really, when we're talking about the area, when we're talking about the effectiveness of something, all we really have to do is to liken it to things that we already know. So population size, right? Okay, species in a population. Okay, women and men sweat different, and so our stuff smells different. We're different species. It's still sweaty, nasty, dirty clothes, but ladies and men, guys and girls, our population sweat is different, and so men's dirty clothes invariably smell different than women's dirty clothes. I would argue women smell worse than men. Ladies, I'm sorry. You guys got whole aisles in the supermarket that have stuff just for you to smell good. Guys, we got like 3 things. Okay, so the concentration of antimicrobial agent, do we need to talk about that?
Yeah.
Okay, well, let me ask you a question. So the concentration is, so I have 10 pairs of dirty clothes in the middle of the room. I can take all 10 pairs of those pants and throw them into the washer right now, or I can take 1 pair of pants at a time and wash them. Which one's more concentrated?
The 10.
Yeah, if I take all 10 pairs at once, my attack or my aggressiveness to get rid of the dirty, stinky smell is more aggressive taking all 10. And so again, the concentration of whatever I'm using, if it's a more aggressive attack, it's most likely more concentrated.
Gotcha.
Okay, the duration of exposure, well, how dang long has those dirty pants been in that room, and how quickly do I get in there and clean them out? Right? If I take 1 pair of pants every minute and put them in the washer, as opposed to taking 1 pair of pants every hour and putting them in the washer, that's the duration time I've exposed the dirty pants to whatever I'm doing to them.
Okay.
Okay. So again, it's just the duration time is okay. So in the case of your dirty dishes, have you guys ever had like dirty dishes that you've let sit for a while and like nasty pasta has stuck, an old spaghetti sauce has stuck to the dishes? How hard is it to get that stuff off of there when you're scrubbing them with your hand?
Hard.
It's hard. So what do a lot of people do? Like me, I actually put water on them and let them sit for a minute. What am I doing when I do that?
Letting it soak in it?
Yeah, the duration of exposure to water loosens up the food particles and makes them easier to get off. I've only got 2 choices. I can scrub harder, which is a concentration, or I can expose them to something that changes their composure and makes them easier to remove. Okay. Then temperature. What happens when we light stuff on fire? It kills it, right? It kills most stuff does not like to be burned up. So we use temperature to affect this stuff. So again, I can use cold water on my dirty dishes, or I can use hot water on my dirty dishes, which is more effective in getting nasty, dirty food off my dishes. So ladies, do you use hot water to wash your dishes, or do you use cold water to wash your dishes?
Hot water.
And why do you do that?
Because the hot temperature kills it faster.
Yeah, it makes it easy. I'll be honest. I'm going to be lazy. I'm going to tell you, ladies, I'm lazy when I clean my dishes. I do hot water, not because I really, in my mind, actively think I'm going to kill germs. I go, the hot water makes it easier to get the nasty food off the dishes faster because I want to go watch Star Trek episodes, man. I got Veronica Mars waiting for me. I got my study group waiting for me. And so it's more a product of just I want to get done sooner. But the second effect is that the temperature also more effectively cleans the surface that we're talking about. And then the local environment, right? So if I open a window and I put a fan on the dirty clothes, forcing the dirty smell out the window, does that environment reduce the amount of nasty smell?
Yeah.
As opposed to I shut the window and I leave those dirty pants in there with no moving air, that room is going to smell like death really quick. Okay, so the local environment can certainly affect the way these antimicrobial agents work, and also how well the bacteria grow. Okay, Here we go. Someone else take this slide. What's going on in this slide? So look at the slope of these 2 lines: high population load, low population load. Is the slope the same?
Yeah.
Okay, what's different then?
The amount, the population.
So does the pathogen or the disease or the bacteria die at the same speed?
Yeah,
Yeah.
Yeah.
But one ends sooner. Why?
because there's
Because it's a
less.
smaller
There's just less of it.
population.
That's all this is showing. My D time is the same. I just have less of something. The D time or the death rate of that thing is the same. It just means that the lower population load's going to die sooner. Real rocket science there, right, ladies?
Yep.
That's really difficult.
Mhm.
Like a lot, absolute rocket science on that one. Okay, I'm done. Who wants to take this slide? Addison, you want to go through this one?
Sure.
This is our patho stuff, man. This is right up your alley. I know we talked about all this stuff in pathophysiology.
Okay, so gram negative then gram positive to disinfectants and antiseptics. They have the outer membrane.
So, Addison, can you explain for Laura and myself the difference between gram negative and gram positive cells in the human body and why which one is more dangerous than the other one to us as humans?
Gram negative has like the 5. Gram positive only has what was it? 3.
Three layers. But remember in pathophysiology, which one slowly releases toxins into the body and which one rapidly like lets the stuff out?
The gram negative rapidly.
Yeah. So, Laura, one of the things you're going to want to remember about gram negative and gram positive for your future for next semester when you take this, gram negative cells, they hold on to their internal components and their internal toxins until they build up a lot of the stuff and then they rapidly release it like a nuclear bomb. Gram positive cells kill you by 1,000 cuts. It slowly releases toxins into the system. And so you're sicker longer, but you're sicker slower. Gram negatives, you'll feel fine, and then they explode and they release a rapid toxic response to your system. And so septic shock is usually a response to gram negative cells.
Okay.
Just that was just a little fun for you just to know, like, hey, when you see that again, you've heard it once and it'll be a little bit easier for you to remember it.
Okay, great.
Okay, cool. Endospores. What's going on with endospores, mycobacteria and protozoans, oocytes?
So
What's
those
going
are
on
the
with those guys?
hardest ones to kill. They're the
What's an oocyte? Does anyone remember what an oocyte is?
Yeah. Oh no. Isn't it like mm,
I think an oocyte, yes. I think you're going the right direction, Laura.
an
Addison,
ovary?
it's a premature like cell that from coming from a woman's ovaries.
Yeah, because an oophorectomy is you have your ovaries
Ovaries
removed.
removed.
Yeah.
Why do you think those particular eukaryotic cells are very resistant to stuff killing them?
Well, for the ones with the ovaries, they're how you reproduce, and so they're like really durable, I guess.
Well, yeah, and also they only want to be penetrated by one thing, sperms, right?
Yeah.
That's you're exactly right, Laura. So they're very, very resistant to everything, but some of the stuff that sperms have on them, some of the acidic stuff that the sperms bring with them, actually are very, very good at breaking down the protection layers of the oocytes. So they're resistant to everything but that. So that was it. I just thought it was kind of fun to just talk about that for a second. Okay, next line. What's the next line say?
non-enveloped viruses are more resistant than enveloped viruses.
Yeah, and that makes sense, right?
What's
So
a non-envelope?
well, I don't know specific non-envelope viruses, but let me ask you guys let me ask you a question. Is it easier to read somebody's mail if it's just if it's a letter sitting on the table, as opposed to an actual sealed envelope sitting on the table? How much harder is it to read that letter with a person knowing it? In a sealed envelope, what do you have to do to get to the message?
Take it out of the
I
envelope.
have to cut the envelope open to get to the message. Well, now everybody knows I cut it open to get in there, right? There's no secret there. It's been cut open and someone else has read it. As opposed to, yeah, I left a message, a letter from, you know, the president of the United States sitting on the desk and it's not in an envelope, it's just a folded piece of paper. How easy is it for somebody just to walk up, unfold it, read it, and fold it back and put it back, and no one know they read it?
Yeah, pretty easy.
So that's it. So again, she said she was going to talk a little bit more specifically about enveloped and non-enveloped viruses later, but just remember, that's the difference. It's just harder to read envelope. Like, you know, people don't go to their next door neighbor and tear their mail open in the mailbox, look at it and shove it back in their mailbox, because everybody would know someone's been in there reading their mail, right? That's why we put stuff in envelopes, kind of to know that people are touching our crap. And then prions, what are prions?
Really hard to kill.
Yeah, they're bacteria with extra stuff around them on purpose to protect them. Okay, moving right along. Okay, what's the most important thing to remember about this?
Isn't a prion a mutated protein?
Yeah, it is.
Okay, just making sure.
And that's mad cow disease, right? So prions are the mad cow disease. So you already have a mutated bacteria in the brain of some organism, right? Well, what do we know about the brain of organisms? They have all kinds of chemical and physical layers keeping stuff out of the brain. So these prions get embedded in there, you know, through the blood-brain barrier. And so now nothing else. We can't send medicines in there to get them because the blood barrier, the brain stops the message from getting to these prions, and those little prions can go crazy. And they're already mutated bacteria, man. They're like super virus. They're like super bugs. Yay, prions.
Okay.
That's gonna be the death of humanity is prions. I'm gonna tell you right now. That's Resident Evil. Okay. So looking what all this shows is what?
That the gram-positive can be killed easier than the gram-negative.
Yep, they're just easier to die. So enveloped, envelope viruses. What the heck? It's saying that what is that right? Okay, it's saying the unenveloped. It's saying the unenveloped are more resistant. Okay, that's the opposite than what I was saying, ladies. You see that?
Yeah.
I was saying the envelope stuff was harder to kill, but I was wrong. So I'm gonna have to make a note of that, because that's kind of counterintuitive. You know what I mean? It's like, wait a minute, why would enveloped viruses be easier to kill? I wonder if that's in the notes later on. I bet you that is.
I don't remember that.
No, that's what I'm saying. That's weird. Okay,
Yeah.
we'll just make a note of that. Envelope viruses are easier to kill than non-envelope viruses. And I'm glad that she put this graph in here, because that makes it a little bit easier to see that. Okay, funguses, active stage protozoas. Yay. Protozoas. Ooh, cysts of protozoas. Ugh. Okay.
Ew.
Nasty. Okay, so control microbial control microbial control methods. So the two main kinds of methods are what? What are the two classes of methods?
Physical and chemical.
Physical and chemical. Okay. So without looking through all these thinking slides, let's go through this really quick. So who wants to take heat and talk about heat?
Me.
Oh,
I
Laura.
will.
Okay.
So you heat it up and it dies.
Okay, so are there some differences?
In what?
We'll just look
How
at what
you
we
heat
have
it?
here. So moist, pasteurization, and dry heat. What are the differences between those guys?
The way it's heated. So some of them are like boiled, and other ones are put in like an oven, and other ones
So,
are
an
under
autoclave.
steam.
What makes an autoclave different than a normal oven? Why don't you have an autoclave in your house?
Probably because it just gets super hot
Well, remember, how does it get super hot?
with the steam.
Yeah, so that's the one where we put the instruments inside of a normal oven, and then we put it under pressure. We lower the atmospheric pressure and expose it to steam. And remember, anytime we put something under pressure, we make it hot. Like, remember how we talked about pumping up the bicycle tire, and the actual tube of the bicycle tire pump gets hot. That's because we're creating pressure, and it's increasing friction and stuff is creating more temperature because of pressure. So the autoclave is a steam with pressure, and that drives up the heat and makes things die easier.
Mm-hmm.
Okay, what's pasteurization? What was the special thing about that one?
So that was discovered by Louis Pasteur.
Yep. But what did he do it on? What was the stuff that he was killing the stuff in? Was he killing medical instrument stuff, death and bacterial on medical instruments, or was he trying to make it safe to
No.
drink milk?
Yeah, he was doing milk and juice and
Yeah.
stuff and drinks.
So why don't we just throw our milk and our juice into the autoclave and click all the stuff that way?
Well, because it would get too
Good
hot
idea.
and curdle it, because the pasteurization heats it, but only below boiling, so it doesn't get as hot.
And does it do it more than once or just one time?
I actually don't know.
Well, if you look at the little graph, it shows those pipes going up and down 3 and 4 times.
So it
So it
doesn't.
heats it up and then it
Oh.
cools and it heats it up and it cools and
Oh
heats it
yeah,
up.
wasn't it like for 15 seconds or something like that?
Yeah. Yep.
Yeah.
Okay, we cool?
Yeah.
Dry heat. What's dry heat? Dry
Is that like
heat's
the
just
oven?
that's just throwing them in the oven. Okay. So Addison, you want to take refrigeration and freezing?
Yeah, so basically they can't become dormant.
Yeah, so let me ask you a question. Do we kill anything with freezing and refrigeration?
No.
We don't actually kill anything. Now, we kill stuff, right? We're actually killing stuff. But refrigeration and freezing, we're not killing anything. We're just forcing them into a dormant stage, right?
Yeah.
Okay, what about desiccation? So desiccation is the absence of something.
Water.
Yep. So in the last chapter, in chapter 9, we talked about water activity, right? So when we remove water from something, when water becomes unavailable, it keeps microbacteria or microbial stuff from growing. So again, similar to refrigeration, I don't think we actually kill anything with we don't actually kill anything with desiccation. We just force it into dormant states, and we make it more difficult for them to grow. We're just changing the environment so it's more difficult for them to grow. They don't actually die. So they talked about simple desiccation is just removing water. So like by adding salt and stuff and glucose, we remove the water by activating it with something else. And then what about freeze drying? What do we call freeze drying?
Lyophilization.
Yep, lyophilization.
Lyophilization, yeah.
Yep, lyophilization. And that's just freeze drying it, right? We expose it to cool air so fast, it just sucks all the water out of it and freezes it.
Yeah.
Yay, fun. Filtration. Filtration is the easiest one, guys.
Filtration just sterilizes liquids and using heat.
Yeah,
So
we
you
use
just
some barrier. We use some
filter.
kind of barrier. The little holes in the barrier. The holes in the barrier are smaller than the size of the particle we want to stop, and it holds all those particles and allows everything else to drain through. That's filtration. Yay, filtration.
Yay. So
Here we go.
I have to go because we have
Okay,
people
it's
coming over
7 past anyway.
for FHE. Sorry, I was on mute. I was eating 4 hot dogs and a salad, so
Just 4 hot dogs and a salad.
that's why I was muted.
Just 4 hot dogs and a salad.
Yeah, I highly recommend putting Chick-fil-A sauce in place of mustard. It's wonderful.
Chick-fil-A. Okay. There's Laura's contribution to our study group.
Sorry.
That's okay. Okay.
Okay, well, tomorrow.
Hey, Addison, are you gonna be there tomorrow with her, or do you have class?
I work till 7.
Okay, that's right. I knew there was a reason you couldn't be there. Okay. Tomorrow at 6 then.
All right.
Later.
Bye, guys.
Okay, bye.
Bye.